Hello and welcome to Nikolai's genetics lessons, and today's problem is assume that the size in rabbits is determined by genes with equal and additive effects from a total of 2012 F2 progeny from a cross between true breeding large and small varieties. 8 rabbits were as small as the small variety and 8 were as large as the large variety. How many size determining genes were segregating in these crosses? According to our problem, we have two varieties of rabbits. One is large and true breeding. Genotype, let's say, would be capital A, capital A, capital B, capital B, capital C, and we cross with true breeding small variety. So let's say the genotype is going to be small a, small b, and small c. We call this first generation, parental generation, so parent 1 here and parent 2 here. And such a cross is going to produce F1 generation, and genotype of the F1 generation is going to be as follows. For the gene A, capital A and small a. For the gene B, capital B, small b. For the gene C, capital C and small c. And we have to cross with the same genotype. So heterozygous for each allelic pair. And in the following F2 generation, we are going to see many different genotypes and phenotypes, and genotype and phenotype of F1 generation is going to be the same. All animals are going to have the same genotype. But in F2 generation, there can be many different genotypes. I will stop here and will show you also different model that is going to explain how we are going to find an answer today. Imagine that we have only one pair of alleles for the gene A. So let's say we cross to animals who is going to be heterozygous. So what we expect in the following generation? Take a look. This is simple Punnett square. One gene, two alleles model. So we are going to have here capital A, capital A, capital A, small a, capital A, small a, and small a, small a. How many phenotypes we have here? Some of you may say that we have 1 and 2 phenotypes here. This is not so. We have 3 phenotypes here because this is additive trait. So each dominant allele would add a little bit to animal height. So this genotype would be different from this, and this phenotype would be different from this. So this is going to be tall, this is going to be intermediate, and this is going to be small. And what our ratio? The ratio is going to be this genotype and phenotype is going to be 1/4. Smallest also would make 1/4, and the intermediate would make 50%. So 1/2. This is one gene, two alleles model. Now take a look at this picture. I want to save your time and I'm not going to draw it. Just probably you know this picture which explains the hybrid cross, where we have two genes, gene R and Y, and each gene represented with two variants of the alleles, dominant and recessive, dominant and recessive. So as you see, two genes, 4 alleles model would give us a square 4/4, which is 16. So one gene, 2 alleles, gave us 2/2 square. And here, with addition of one gene, we now have 4/4. Please pay attention that here on this picture, we see that for example, genotype capital R, capital R, capital Y, phenotypically the same as R, Y capital and Y lowercase. But if trait is additive, these two genotypes are going to be different phenotypes. Let me explain one more time. For example, if dominant allele would add 10 centimeters to the height of the rabbit, so this genotype would produce rabbit that is going to be 40 centimeters tall. And let's say that recessive allele add just 5 centimeters. So here we are going to have animal which is going to be 35 centimeters tall. And this animal also going to be 35 centimeters tall. And this animal is going to be 30 centimeters tall. So this is going to be different phenotype from, for example, this phenotype. So this is going to be different phenotypes. But if we have dominant and recessive allele in simple Mendelian genetics, this is going to be the same phenotype. But again, not the same phenotype if we consider that this trait is additive. And we know from our problem that effect of the genes are additive. So again, this genotype is not the same as this. So genotypes that produce animal of the maximum height, as you see, would be 1/16. Only one such genotype here where all 4 alleles are dominant. You cannot see any other such genotype here, and only one genotype here which produce, in our case, for example, if it's going to be animal height, animal with minimum height, and it's also going to be 1/16. Now again, take a look. Here it was 1/4 and 1/4. Here it is 1/16 and 1/16. So now I think you can follow this logic, and we can build a simple table. Take a look. For example, with a model where we have one gene and 2 alleles, we are going to have a Punnett square which is going to be 2 by 2. So this is, I am describing this model, one gene, 2 alleles. And extreme phenotypes, extreme tall and extreme small, is going to be 1/4 and 1/4. Extreme tall and extreme small. Now let's take another model, 2 genes, and we are going to have 4 alleles. And our Punnett square is going to be 4 by 4, as you remember. I just show you the picture. And the extreme phenotypes, extreme tall and extreme small, is going to be 1/16. 1/16. Now let's take another model with 3 genes. So 3 genes. I didn't show you, but we just would follow this logic. 3 genes are going to produce 6 alleles. And our Punnett square is going to be 8 by 8 cells. And extreme phenotypes are going to be, take a look, this number is 4 times smaller than previous number. We are going to have here 1/64 and 1/64. Now let's take a model with 4 genes, so 4 genes, and this time we are going to have 8 alleles, and our Punnett square is going to be 16 by 16 cells. And extreme phenotypes are going to be 1/256, and extremely small would be 1/256. So this is 4 genes and 8 alleles model which produce such phenotypes. Again, extremely large would be about 1 out of 256 animals, and extremely small would be 1 out of 256 animals. And all the rest, 254, would be intermediate between these two extreme phenotypes. So would be intermediates. Now let's return to our problem. We are told that in F2 generation we got 2012 animals. 8 of them, 8 rabbits, were as small as the small variety, and 8 were as large as the large variety. So what do we have? We have a ratio 8 extremely large to 2012 total number of animals to 8 extremely small. Now let's simplify this ratio by dividing all numbers by 8. And we are going to have a ratio 1, 251 here, and 1 here. So our new ratio is going to be 1 to 251 to 1. And we can say that this extreme genotypes here and phenotypes is going to be about 1 out of 251 animals. 1 out of 251, you see 1 divided by 251. We also can say this is the same as 1 over 251. So it is very close to this number. We do not expect that number is going to be exactly as here, because this is just approximation. We have found that if we have extreme phenotypes as 1 out of 256, we have a model with 4 genes. So 4 genes and 8 alleles. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.